Hello, everybody. My name is Nathaniel Haskin. I'm a wildlife biology major here at NC State, and today I'm going to be talking to you about my Snapshot USA project. And what my group did was strictly focus on protected areas. And with this idea in mind, we came up with two different hypotheses. Both are very similar, but are slightly different in ways as well. Our first one was talking about mammal abundance and does it increase or decrease either the farther you get from the protected area, like just farther away from it in general, or the closer you get to the protected area, not necessarily inside, but like right outside of it. And then our second hypothesis talked about does mammal abundance either increase or decrease inside the protected area, either closer to the core, at the very middle of the protected area, or does it increase or decrease closer to the outside boundary between inside and outside? So what we did is we took the data of a lot of different observations of mammals all across the country, and we had three main groups in mind, and we were mainly focusing on detection rates, both from people and from like camera traps and trail cams and stuff that caught animals in certain locations where they were known to be around a lot. Now, why we did this was it helps scientists know where certain species are, and this is especially useful information to know with endangered or threatened species that have very low numbers. And it also just assists both biologists and scientists so that we know what populations are doing well and which ones aren't, and how we can keep the healthy populations healthy for longer, and how to increase the populations for the numbers that are dwindling so that those populations become stable over time. Now, we also compared big protected areas versus small, and both inside and outside for both groups. Now, inside the protected area, both sizes, it doesn't seem to matter. The numbers are very, very close together, only about a 0.013 difference, versus being outside the protected area, the numbers are difference is a little bit higher. It's closer to 0.2. Now, with this graphic, if you look on the left, this is the graphic for all of the mammals that we looked at. And as you can see, the small and big protected areas, there were several outliers in both locations, both about maybe the middle of the data. And the small protected areas usually have higher average numbers right before and right after the big areas, with the big areas taking up a big chunk of the middle. And then the graphic on the right is very similar, showing almost the same type of distribution, but this is strictly for white-tailed deer. And as you can see, the big protected areas usually have higher numbers in the middle, with the small protected areas having higher numbers on the right and left side of it in terms of distance. So a little bit closer would be the smaller areas, and same with a little bit farther as well. And so for the results that we found that of our three groups, which our three main groups were large carnivores, small mammals, and ungulates, which is shown on this graphic in that order. So blue would be large carnivores, yellow and orange would be small mammals, red would be ungulates. What we found was that large carnivores had the lowest detection rates. Group 3, which was the ungulates, had the intermediate values, and then group 2, which is the small mammals, had the highest detection rates. And this makes sense because the larger the animal, the less of them there are, which means the smaller the animal, the more likely you are to have more detections of them, because there are more of those small mammals around. And what we found, like what I just said, was the smaller the mammal, the higher number of detections that occurred. And this seemed to happen in both large and small protected areas, and both inside and out. And there were higher numbers of detection rates for especially the small mammals that occurred both inside and outside protected areas. So that's it for my presentation. I hope you guys enjoyed, and thank you for your time.